So in this chapter, we're going to look at genetics. And when we get to practice problems, you'll want to pause your video and try and work out the practice problems yourself and then push play when you're ready. So an overview of what is covered in this chapter. We will start by talking about Mendel's law and talking about the basics of genetics. Then we'll move on and look at variations in Mendel's law. So we'll talk about things like pleiotropy and environmental influence and polygenic traits, etc. We'll look at the chromosomal basis of inheritance, and then we will talk about sex chromosomes and sex-linked genes. So we'll start out with Mendelian genetics. Gregor Mendel was the first person to comprehend some of the most basic principles of genetics, and he reached these understandings in the mid-19th century, working in what is now the Czech Republic, and using his experimental subjects, a species of garden pea, which you can see below. So here we have these purple flowers and these white flowers, and so he was a person who really studied genetics. So Mendel discovered the principles of genetics in experiments with the garden pea, and he showed that parents pass heritable factors to the offspring. And those heritable factors are what we now call genes. Now, advantages of using pea plants. 1, controlled matings. So he could control when matings occurred. Think about if you were trying to study genetics in humans. Not easy. You can't tell two people that they need to reproduce, right? But in pea plants, he could control the matings. He could do self-fertilization, so the plant could pollinate itself, or cross-fertilization. The traits that he was looking at were observable characteristics with 2 distinct forms, meaning purple flowers or white flowers, or green seeds or yellow seeds, or round seeds or wrinkled seeds, etc. So this is obviously in contrast. Think about humans. Are there only 2 eye colors? No. Are there only 2 hair colors? No. That gets a little more complicated. He had true breeding strains, which means that the same trait will be passed on with every self-cross. So those white flower plants would always give rise, when self-fertilization, to white flower plants. So Mendel crossed pea plants that differed in certain observable characteristics or traits, and then traced the traits from generation to generation. And so this illustration that you're seeing on the right shows his technique for cross-fertilization of 2 types of flowers. So we'll call the purple plant the mom. And so he took the mom plant and he removed the stamens. Stamen, think men, male part. That's the male part of the flower, so that that purple flower plant couldn't self-fertilize. He then took another plant, in this case we'll call it the dad plant, the white one, and he transferred sperm-bearing pollen from the stamen of the white plant and put it on the female reproductive organ on the mom plant. So basically he's helping fertilize from one flower to the other. The pollinated carpels then mature into pods. They plant the seeds from the pods, and then he would look at the offspring to see what traits he saw. And so this allowed him to track how traits are inherited. So Mendel studied 7 pea characteristics. So again, we have flower color, purple versus white, position of the flower, axial, meaning along the middle of the stem, versus terminal at the end, seed color, yellow or green, seed shape, round or wrinkled, pod shaped, inflated or constricted, pod color, green or yellow, and then stem length, tall or dwarf. And he hypothesized that there are alternative forms of a gene, although he did not use that term, and that these alternative forms of gene are the units that determine hereditary. So we now call these alternative forms alleles. So alleles are these alternate forms of a gene that will give you potentially different physical characteristics. So again, alleles are alternative versions of a gene. A gene is found at the same loci, right, or locus, singular, on both homologous chromosomes with one allele on each. So again, if you think back to meiosis, when we talked about homologous pairs, remember that homologous pairs are matched in size, banding pattern, and the genes. So notice that if we look at these two chromosomes, these are a homologous pair. One came from mom, one came from dad. And so we have the locus for flower color gene. Now, at that loci, at that position, we may have alleles, alternate versions of a gene. So we could have a plant that has the allele for purple flowers on one and the allele for white flowers on another. They both code for flower color, yet they don't give the same outcome. So when we look at alleles, we can have what are called a dominant allele, which we give a capital letter, or a recessive allele, which gives a lowercase letter. In the case of flower colors, the purple allele is dominant over white. So when we give those alleles a letter, the dominant is going to be capital and the recessive is going to be lowercase. And so I notated that on the image. So big P for purple flowers, that's dominant, over white flowers, which is recessive. Certain letters, P's, S's, C's, where the uppercase and the lowercase look very similar, you want to make sure that when you're writing those out that you have some way to distinguish between whether it's an uppercase or a lowercase. For me, on my lowercase p's, I put this little like curve at it, and then I know that that's my lowercase letter. So let's talk about what does it mean when an allele is dominant. So the dominant allele is the allele that is expressed in the heterozygous. So in the case of purple and white flowers, we would say that that gene is heterozygous. That'll be on the next slide. Hetero means other. They have two different genes, two different alleles. So the dominant one is what we actually see. So in the case of the flower colors, if the plant has a purple flower allele and a white flower allele, what we're going to see is going to be purple flowers. The purple allele is dominant over the white. That's the one we actually see. The recessive allele is the white flowers. This is the allele that is not expressed in the heterozygous. So we don't see the plant having white flowers, but it doesn't mean that that allele isn't there. It's just not being expressed. It's not being seen in the physical characteristic of that plant. And so this would be alleles. So if we look and we call something homozygous, that means that they have the same allele. So the homozygous will have the same allele. So notice that if we look at these homologous pairs, one is from mom, one is from dad. Notice that on the one from mom, it's a big P. On the one from dad, it's a big P. Therefore, this is homozygous. It's the same allele. Just think of like homosexual versus heterosexual. Homosexuals like the same sex. Heterosexual, other. So the homozygous is going to be the same allele. So you could be homozygous for the dominant allele. Notice that this would be big P, big P. You could be homozygous for the recessive allele. This one is going to be little a, little a. Or there could be heterozygous. Hetero means different. So big B, little b. Notice that those alleles are different. And so that would then be heterozygous. So when Mendel did his experiment, he deduced that an organism has two alleles from each characteristic, and that one allele comes from each parent. The alleles from the parent may be the same or they may be different. So what he did was when he took his parental generation and he had his true breeding plants, he had a purple flower plant and a white flower plant, meaning that when those were able to self-fertilize, the purple flower plant always gave rise to purple flowers. and the white flower plant always gave rise to white flowers. When he crossed them, he found that all of the plants had purple flowers. There wasn't a blending event. It wasn't that the flowers were in between. They weren't lavender. They were purple, just like one of the parents. However, when he took those purple flower plants that were the offspring of the perennials and he crossed those, what he saw was that he got 3/4 were going to be purple flowers and 1/4 were white flowers. And so notice that the white allele, that white gene, didn't disappear. It wasn't gone. It just wasn't seen. And when we cross that F1 generation, now we see the offspring having white flowers. And so this happens all the time. You could have, for example, two parents that have brown eyes and they have a child that has blue eyes. That's because both parents carried the blue-eyed allele. You didn't see it, but when they came together, by chance, the offspring ended up having blue eyes. So these traits are not gone. They're just not seen. And so here's a perfect example of this. This is a biracial couple, and they have twins. You can see in the baby's faces that their structure in their faces is actually quite similar. However, their coloration is very different. One is much more fair, has light color eyes, light hair. The other twin is darker skinned. That's because of simply random chance. The baby who is lighter skinned happened to, just by chance, inherit both of her parents' lighter coloration genes. And therefore, she is lighter in coloration. And so this just shows that alleles are retained across generations. Even if we don't see them in the parents, it doesn't mean that those alleles are not present. They're there, they're just not seen. So when we look at Mendel's experiment, when we look at the purple flower plant, if it is true breeding, that indicates that it is homozygous. Homozygous, remember, big P, big P. Now, when we looked at meiosis, remember that when we see big P, that means that that allele is found on the homologous pairs, one from mom and one from dad. So in that purple flower plant, homologous pairs are going to separate during meiosis and during the formation of gametes, so that the gamete is only going to have one letter. In the case of the purple flower plant, the only gamete that is possible is going to be the big P. That's all that parent can give. The white flower, on the other hand, the white flower, white is recessive. The only way that you can have a white flower plant is if it is little p, little p. So that white flower plant has its little p allele to give. It's the only allele it can give, so its gamete has a little p. So purple flower plant gives the big P, white flower plant gives the little p. When they come together, when those gametes fuse during fertilization, and they restore diploid number, now the offspring are going to be big P, little p. That will give us a phenotype, which you'll see in a minute, a phenotype of purple flower plants. They will appear purple because the presence of the big P, the dominant allele, is going to give us purple plants. So now when we take those purple flower plants, this what we call F1, filial 1 generation, and we cross those with themselves, notice that that purple flower plant has big P, little p. It is heterozygous. So it has a probability, a 1/2 chance of giving big P and a 1/2 chance of giving little p. It's a 50-50 probability. It all depends on the way that the chromosomes line up during meiosis. Now, a sperm or egg only carries one allele of each pair for a given gene. So again, the sperm is only going to have one allele. It's either going to carry the big P or the little p. And those pairs of alleles segregate or separate when the gametes form. And this process describes what is called Mendel's law of segregation. So he hypothesized that those traits, those alleles, will separate during this process. Now again, the alleles could be dominant or recessive. So we need a few definitions. So the P stands for the parental generation. The F stands for filial or offspring generation. So the F1 are the offspring of the parentals. The F2 are the offspring of the F1s. Genotype. Genotype are the two alleles present in an individual's for a gene. So the genotype basically is what genes does that organism have. Phenotype. Phenotype is going to be the physical result of having a particular genotype. So if we look at an example of genotype, a genotype would be what genes, so what alleles. So a genotype could be big P, little p. That's its genotype. Its phenotype is what you're physically going to see. So if the plant is big P, little p, that means that its phenotype is going to be purple flowers. That's what we actually see as a result of having big P, little p. So question for you. In Mendelian genetics, an individual has the following genotype for a specific gene. Big A, little a. Molecularly, this means that is it red, the big A and little a alleles are at different loci on the same chromosome. So are they on the same chromosome but different positions? Yellow, the big A and little a alleles are at the same locus on sister chromatids. Or green, the big A and little a alleles are at the same locus on homologous chromosomes. So pause your video, and when you're ready, go ahead and push play to hear the answer. So if you answered green, you are correct. The big A and little a alleles are at the same locus on homologous chromosomes. So it can't be red. Red says that the big A and the little a allele are at different loci on the same chromosome. They're not on the same chromosome, but instead they're on the homologous pair. The genes, if they're a gene, they have to be at the same loci for them to be the same gene. So they are going to be at a particular position on the chromosome, and they can't be together on the same chromosome. Big A, little a alleles are at the same locus on sister chromatids. You can't have a situation where they're going to be on sister chromatids. So if we have these two chromosomes, notice that one has the big A, the other has the little a. Those can't be sister chromatids, because remember that sister chromatids are exact copies of one another. So if I were to draw a sister chromatid for the big A, on the sister chromatid, it's also going to have the big A, because those sister chromatids come about by DNA replication. They're going to make an exact copy. So yellow doesn't make sense because they can't be on the sister chromatids. Sister chromatids are identical. If it has a big A on one sister chromatid, it has to have the big A on the other. So it has to be green. They're at the same loci on the homologous pairs, one from mom and one from dad. And so you want to keep that in mind when you're thinking about the genetics, right? Because remember when we talked about meiosis, homologous pairs are going to separate so that the gametes are only going to have one of those two chromosomes. Either they're going to give their offspring the big A allele, or they will give their offspring the little a allele. So when we talk about a monohybrid cross, mono refers to one. We're looking at one gene. And so we can use a Punnett Square to determine the types of offspring and in what particular ratios, meaning what is the expected outcome. So if we look at our F1 generation and we cross our F1s, remember that the genotype is big P, little p. So on the outside of our Punnett Square, we'll put the possible alleles that they could give. So if we have big P, in the gametes, the probability is 1/2 that we give a big P, 1/2 that we give a little p. That's by independent assortment. So notice that there's a chance for the sperm to have big P or little p, and there's a chance for the egg to be big P, little p. Because remember that we're crossing two F1s. Both are big P, little p. So you see on the outside of the Punnett Square, the possible alleles that could be given in the gametes. In the middle of the Punnett Square, what we fill in, if you notice this one here, we have big P. So the sperm gave the big P, the egg gave the big P, we get big P. That is a possible genotype for the offspring. Or the egg could give a big P and the sperm could give a little p, so that the offspring's genotype is big P, little p, which is still going to be purple flowers. However, if we look at the egg giving a little p and the sperm giving a little p, then the genotype is going to be little p, and the offspring are going to have white flowers. So there is an expected ratio of 3 to 1. We would expect that if these two F1 plants mate, 3/4 would be purple and 1/4 would be white. And so one of the advantages of using pea plants is that he could produce a large number of offspring, so that when he did these experiments, he would see these expected ratios. That doesn't always happen in the case of humans. We have so few offspring that it's by chance not necessarily going to work out to the correct probabilities. And what I mean by that is if you think of gender, for example, sex. So sex, parents, when they have kids, they have a 50% chance of having a boy and a 50% chance of having a girl, right? And that's because you're going to see that the males will determine the sex of the child, because females are XX. Females are always going to give their kids an X chromosome. The males are XY. So there's a 50% chance that he gives the X, and there's a 50% chance that he gives the Y. And so depending on which one fertilizes the egg, that's going to be the sex of the child. Now, if you think about, people you'll know that it does not mean that even though the probability is 50-50, that doesn't necessarily mean that that's what's going to happen, right? You've probably seen some families. I have a friend who has 4 boys. That's not the expected ratio. Out of 4 kids, you would expect 2 would be girls and 2 would be boys. But in fact, just by sheer chance, they ended up with 4 boys. So that has to do with our low number of offspring. Because we have a low number of offspring, we don't necessarily always see the expected ratio. The more offspring you look at, the more likely that those probabilities will match what happens in actuality. And so the same thing can be said with a coin. You could simply toss a coin 4 times and it could end up heads every time. But if you flip that coin 1,000 times and your sample size is a lot bigger, well, then the probabilities are more likely, the 50-50 is more likely to match what is seen in actuality. And so the pea plants were a good system because they give large number of offspring. So when you work on monohybrid crosses, some keys to kind of help you along the way, and I will show you an example problem. So how do you want to attack these types of problems? 1, write down the phenotypes and determine the genotypes. So for the parents, you want to write down the phenotypes and the genotypes. Then you want to figure out the number of different gametes that can be produced. You want to set up your Punnett Square and place the parents' genotypes on the outside. So the gametes that the parents could produce will go on the outside. And then determine the genotypes and the phenotypes of the offspring by filling in your Punnett Square. And so let's look at an example. So in pea plants, tall stems are dominant to short stems. When a homozygous tall plant is crossed with a homozygous short plant, what is the probability that the offspring will be short? So when I solve these types of problems, I like to start by writing out what is dominant and what is recessive. So tall stems are dominant to short stems. So I'm going to write big T, some second allele. It could be big T, or big T, little t, is going to give us tall. And short, the only way you get short is little t, little t. So we cross a homozygous tall. So I like to start by writing the phenotype first. Homo tall is crossed with a homozygous short. So I start by writing my phenotypes first, and then based on that information, I will fill in the genotypes. So homozygous tall, homo means same allele, tall is dominant. So this has to be big T, big T. So homozygous short is going to be little t, little t. Now, did I have to tell you that it was homozygous short? Did I have to give you the part that says homozygous? The answer is no, right? Because if the plant is short, you know it has to be homozygous. The only way you can see the recessive trait is if they are in fact homozygous recessive. So I didn't have to give you homozygous short, but I did anyways. Now, based on this, you need to figure out what are the possible gametes. So homozygous tall, there's only one possible gamete, big T. That is the only possible gamete because they have big T, big T. They're always going to give the big T allele. The homozygous short can only give the little t. So when I fill in my Punnett square, I need to put one parent on one side and the other parent on the other. So notice that I can do big T here, little t here. I don't have to write little t, and big T, big T. You can if that makes you feel more comfortable, but you'll see why it doesn't matter in a minute. So I put my gametes on the outside. Now I fill it in. Big T, little t, big T, little t, big T, little t, big T, little t. And so notice that in every case, there's 100% probability that the offspring will be big T, little t, which means that the phenotype would be tall. We would expect that all of the plants would have tall stems. Notice I didn't have to do this other Punnett square part. I didn't have to do this part or this part or this part because it gave us the same thing. So you can simplify. I like to simplify and I just do the ones that are needed. So I just write one parent can give big T, the other parent can give little t, and you only have to fill in one box. So the offspring, there's 100% probability that the offspring are going to be big T, little t, which will give tall offspring. So the probability that the offspring are short, that's a 0% probability. There's no chance of having short offspring. All of the offspring are going to be tall. So here is a practice problem. For this one, you are going to post your answers for the odd numbered practice problems. So there will be 3 practice problems that you have to put your answer. Practice problem number 1, so this one. Practice problem number 3, and practice problem number 5. You will put your answers to those on Canvas on a discussion board. So let me read the question. Round seeds are dominant to wrinkled seeds. When a heterozygous round seed plant is crossed with a homozygous wrinkled seed plant, determine the probability that the offspring will form wrinkled seeds. So I want you to attempt this problem and then post your answer on Canvas. What did you get as your probability that the offspring will be wrinkled seeds? So the question is, can you always tell genotype from phenotype? And the answer is no, right? If you see an organism with the recessive phenotype, in that case, you do know its genotype. It has to be the homozygous recessive. In the case of flower colors in plants, the pea plants, white flowers is the recessive phenotype. And what that means is that the white flowers we know have to have a genotype of little p, little p. It's the only way you get the recessive phenotype. On the flip side of this, if we have a dominant phenotype, meaning a purple flower plant, just by simply looking at them, there's no way to know its genotype. In order to determine its genotype, we need to do what's called a test cross. And so that unknown purple flower plant could be homozygous dominant, it could be big P, or it could be heterozygous, big P, little p. So what we do in a test cross is we're going to cross the unknown dominant phenotype. So we're going to cross this purple plant with the homozygous recessive because we know its genotype. And we're going to look at what the offspring would be. Now, if the white flower plant is the sperm, that is going to be little p, little p. Those are the types of gametes that it could produce. If the unknown purple is big P, that's the example on the left here, big P, then all of the offspring are going to be purple because if it's homozygous dominant, that means that all of the offspring are going to get that dominant allele. If on the flip side, if the organism is heterozygous, it's big P, little p, What that means is that for the gametes for that one, it's going to be big P or little p. If that's the case, then you can see white flowers, because white flowers means that you get a little p and a little p from both parents. So notice that the probability would be that you get 50% purple, 50% white. Now, if you only had a few plants to do this with, and you saw any white flower plants, would that tell you, even if it wasn't 50%, would that tell you the unknown genotype of the dominant? Answer is yes. If you saw even one white flower plant, that tells you that the purple plant had to be heterozygous, because that is the only way that you end up with offspring that are homozygous recessive. And so the same thing can be true if you look at human traits. If you had a dominant trait, for example, you couldn't know what your genotype was without more information. Now, if on the other hand, you have a child and that child has the recessive phenotype, well then yeah, you would know you're heterozygous. Or if you had a parent who had, let's say we're talking about eye color. If you had a parent with blue eyes, you know that you are heterozygous. So in humans, obviously we can't do a test cross. We can't tell people to mate with somebody else. But we can trace it through family trees to try and figure out those unknown genotypes. Mendelian inheritance follows the rules of probability. And that is, if you think about tossing a coin, right? If I were to flip a coin, I have a 50% probability getting heads and a 50% probability of getting tails. Now, let's say I flip once and I get heads. Does that mean that when I flip the coin again, that I'm going to get tails? Answer is no, right? The outcome of that first flip has no effect on the second flip. So while we expect to have a 50-50 probability, each flipping of the coin is an independent event. It happens independently of what happened before. Same thing when talking about genetics. Just because you had a boy, let's say, the first time, does not mean that you're going to get a girl the next time. The outcome of sex in the first child has no bearing on what the second child is going to be. We call this independent assortment. They are each independent events. So if you think about gametes, right? If you have big B, little b, there is a 50% probability of giving big B and a 50% probability of getting little b. And the same applies to the sperm. So when you think about probability, while we expect certain ratios, that doesn't always happen because each event is an independent event. So what we had been talking about before were what we call monohybrid crosses, looking at one trait. But now we want to talk about what would the result be from a dihybrid cross. So if we cross two genes. And so the answer is, do these characters travel together in heredity? Meaning, are they linked to one another, or do they travel separately? If you get one character passed on to a plant, do you definitely get the other one too? And so we're going to look at some different approaches and talk about our dihybrid crosses. So what we're looking at is what Mendel called independent assortment. So we're going to go through two different hypotheses for how these genes are passed down. So in the dependent hypothesis, this is that the alleles for one gene are linked to the alleles for another gene, so that a heterozygous parent can only produce gametes of one type or of two types. Remember that when Mendel was doing this, he did not know about the existence of DNA or chromosomes. So the idea of genes that may or may not be on the same chromosome was not something he could have considered. He just wanted to know, were those traits linked? So let's walk through this. So if we start with a plant that is big R, big Y, big Y. The R is for round. The round seeds are dominant over wrinkled seeds, and yellow seeds are dominant over green. So in this case, big R, big Y, big Y. This is crossed with little r, which is going to be a wrinkled seed, and little y, which is a green seed. So in the dependent hypothesis, that is basically saying that these genes are linked. So what that means in actuality would be that those two genes are on the same chromosome. So this is what the chromosomes would look like. So if they're linked, the big R and the big Y would be on the same chromosome, and they would have 2 of those chromosomes. So what that means, though, is that the gamete definitely would look like this, where it would have a big R and a big Y. If we look over here at this one on the right, the gamete has to be little r, little y. So 100% of the offspring would be big R, big Y, little y. So the F1 generation, the way that the big R, little r would be arranged and the big Y, little y would be arranged is that one chromosome has the big R and the big Y together, and the other chromosome has the little r and the little y together. And so notice that that makes it heterozygous, but that there are only 2 possible gametes that can form. Either they're going to give the yellow chromosome or the green. So if we look at a Punnett Square, let's look at what the outcome would be. So the possible gametes would be either that they give the yellow chromosome or the green. And same thing for the egg, the yellow chromosome or the green. So if the 2 come together, the 2 yellows, that would be big R, big Y, they would be yellow and round. If we have the yellow with the green, it would be heterozygous, big R, big Y, little y. Same thing with down here. That one would also be heterozygous, big R, big Y, little y, and 1/4 would be the green and wrinkled. Notice that there was no mixing of those chromosomes, meaning you never saw a round green and you never saw a yellow wrinkle. So if the dependent hypothesis is correct, you would expect that you would get 3/4 would be yellow and round, 1/4 would be green and wrinkled. But this is not what they saw. And so in the case of the genes that Mendel was tracking, they in fact were not dependent. They weren't on the same chromosome, but instead were inherited independent of one another. And so that's going to be the independent hypothesis. And so this is just drawing out those 2 options so that you could see them by chromosomes. So yellow round, again, the original, the P generation, big R, big Y, big Y. First generation, the parental generation, we have wrinkled and green, little r, little y, little y. When they have offspring, the yellow is going to be given and the green is going to be given. So what you get is that the F1 generation will be heterozygous. It'll be big R, big Y, little y. And the same would be true for both egg and sperm. So they would have these sets of combinations. Now, when you cross those F1s together, this is just to show you how the chromosomes would be inherited, but let's draw out what the options would be. And so our Punnett Square would look like this, where we have one parent giving either the yellow or the green, and the other parent giving the yellow or the green. And so now if we look at the options, we just need to fill in our Punnett Square. And so what you would get is that these 2 parents could both give the yellow. That would be a round and yellow. This parent could give the yellow. This parent could give the green. That is still round and yellow. This parent could give the round, and this parent could give the green, round and yellow. And the last one would be to get 2 of the green chromosomes. So again, you would expect to see 3/4 of the offspring would be round and yellow. 1/4 of the offspring would be wrinkled and green. And that's the dependent hypothesis. This is not what Mendel saw, though. He did not get this expected ratio, which showed that these genes are not dependent. They are actually independent. So if the alleles assorted independently, that's referred to as the independent assortment or the independent hypothesis. And this says that the alleles for one gene are not linked to the alleles on the other, so that the heterozygous parent can produce gametes of 4 types. Remember, that's different than in the dependent. In the dependent, there were only 2 possible gametes, but when they assort independently, there are actually 4 types of gametes. We'll come back to this. Let's take a look at what those gametes would look like. So let's take a closer look at the independent hypothesis. And so that is assuming that the alleles are going to assort independently. And so what you see here is that these chromosomes are arranged by homologous pairs. So here is a homologous pair with this one. Notice they're the same size, same centromere, etc. This one and this one are a homologous pair. So in the heterozygous, the big R, little r, what you're looking at is that these two blue ones both have a big R. Because remember that these are sister chromatids, they are exact copies. So one parent gave this homologous pair and this homologous pair, which is why these are in blue, and these are green. This came from the other parent. So this is how the organism became heterozygous for the F1. It inherited this blue chromosome and this green. Now, remember that we talked about that one of the ways in which meiosis generates diversity is that through independent assortment. And so what that means is that each homologous pair can line up independently of any other homologous pair. So if we look at one possibility, we could have the two blues line up and the two greens line up on the same side, so that in meiosis 1, when these homologous chromosomes separate, the blue ones all go here, the green ones go here. When we get to meiosis 2, sister chromatids are going to separate. And so notice that from this possibility, there are two gametes, big R, big Y, little r, little y. However, it is completely possible that we get this arrangement instead, where we have the blue one on this side and the green one on this side, so that the offspring in that case would be big R, little y, the gametes, and this gamete would be little r, big Y. So if you remember back to our dependent hypothesis, there were only two types of gametes. The only possible gametes was the big R, big Y, and little r, little y. However, because these are on separate chromosomes, they can line up independently. And so it's entirely possible that you get big R with little y, and you get little r with big Y. So notice that with the independent hypothesis, that suggests that these genes assort independently, and that's because they're on separate chromosomes. There are now 4 possible different gametes. And so let's go back and look and see what is the outcome of having these 4 possible gametes. So if we look at our 4 possible gametes, remember that that can come from both parents. So here are my 4 possible gametes: big R, big Y, little r, big Y, big R, little y, little r, little y. And that's the same for both the egg and the sperm. So now if we were to fill in our Punnett square like this, notice that our probabilities are different than the dependent hypothesis. In the dependent hypothesis, remember we expected 3/4 yellow and round and 1/4 green and wrinkled. But notice that when those chromosomes assort independently, because they're on separate chromosomes, we now have different options. We have a probability of getting 9/16 yellow and round, 3/16 green and round, 3/16 yellow and wrinkled, and 1/16 green and wrinkled. Notice by the independent hypothesis, we have these new combinations that weren't seen before. Before it was either always yellow and round or green and wrinkled. But now, because these genes assort independently, we get more diversity. We can have green and round and yellow and wrinkled. And this is actually what Mendel saw when he did his experiments. He saw that these genes assorted independently. Now, does that mean that all genes assort independently? No, because if they are on the same chromosome, and especially if they are close together, then those traits won't assort independently. But in the case of what Mendel was looking at, his traits did assort independently, and that's because they were on separate chromosomes. So when we look at genetics, we talk about probabilities, and that is the likelihood that an event will occur. We have what's referred to as the product rule, and that is the probability of simultaneous independent events equals the product of their individual probabilities. So this would allow you to predict the chance of parents with known genotypes to produce offspring of a particular genotype. So if we were to consider the probability of obtaining a plant with wrinkled, green peas, so genotype little r, little y, from dihybrid parents. So if we have these dihybrid parents, and they are big R, big Y, and we wanted to calculate the probability of getting wrinkled and green, we could treat those as independent events and then multiply those probabilities together. So this is an example of this. So here we have our heterozygous individuals, big R, big Y. If we want to know the probability of getting little r, little y, you just treat them as separate crosses. So you cross just the R's. So notice big R, little r. Treat that as its own separate cross. So we have big R and little r, and then big R and little r, and we fill it in. And notice that the probability of getting little r is 1 out of 4. Do the same thing for the Punnett Square for the Y's. Cross big Y, little y, and look for the probability of getting little y, which is 1/4. So if I want to know the probability of getting both little r, and little y, those are two independent events. They don't dictate each other. So we just multiply those fractions together. 1/4 times 1/4. Multiply the numerators, the number at the top. 1 times 1 is 1. Multiply the denominators together. 4 times 4 is 16. So the probability of getting offspring that are wrinkled and green is 1/16. And you also saw that in the big Punnett Square. You saw the exact same probability. So the product rule is just a shorthand way to solve genetic problems without basically making that large Punnett Square. This for me is easier. If you like the method using the big Punnett Square, that is fine. I'll go over both when we go through an example problem. But for me personally, I know that I get less confused if I just treat them as separate crosses. I cross the R's by themselves and I cross the Y by themselves. And then I multiply those two probabilities together. So this is just an example showing how you can use this to track 3 traits, for example. So if we are tracking a metabolic disorder that causes people to have red urine, that is recessive. So if we have just normal urine, big B, little b, that's normal yellow urine. Urea, which is red urine after eating beets, would be little b, little b. Big H, little h gives you colored eyelids. Little h gives you normal eyelids. Big E, little e gives you short fingers. Little e gives you normal fingers. So let's say that we wanted to cross these two parents. So one parent is big B, big H, little e, crossed with a second parent that is little b, little h, big E, little e. And we want to calculate the probability that a child has red urine, is heterozygous for colored eyelids, or just has the colored eyelids, and has short fingers. So what you do is you're just going to cross the B's on their own. So again, big B, little b, little b. So here's our Punnett Square for the B's. Big B, little b, little b. Fill it in. Probability of getting little b is 1/2. Do the same for the H's. Cross just the H's. So big H, little h, crossed with little h. Probability of getting colored eyelids is 1/2. Then you can cross the short fingers. So little e, crossed with big E, little e. So you fill out your Punnett Square, little e, big E, little e. Notice that for each of these, the probability of each independent event is 1/2. So you have 1/2 times 1/2 times 1/2. So you multiply the ones across the top, that's 1. And then you multiply the 2's on the bottom. So 2 times 2 is 4. 4 times 2 is 8. The probability of getting those 3 would be 1 out of 8. So again, to me, that is much easier than trying to fill in a very large Punnett Square. And all you have to do is treat them as independent events, which they are, and then just multiply your probabilities. So here is a practice problem. An individual with the genotype big A, big A, big B, big C, big D, can produce how many different types of gametes, and what are the possible gametes? So I'm going to show you a couple different ways that you can solve this problem. The first is to just simply solve how many different types of gametes can you produce. So the easy way, if it's just asking you the number of different gametes, you just take 2 to the n power, where n is the number of heterozygous pairs of alleles. So if we look at this example, when it's big A, that's not going to lead to different gametes, right? The A's are both homozygous, so that means that the offspring will always have big A. If we look at the B's, notice that the B's are going to be our heterozygous, so we'll circle this. That's a heterozygous pair. We have C's, which are heterozygous, so they could give either to their gametes. And then big D is homozygous, so that doesn't give different gametes. So if I just need to know the number of different gametes, there are 2 heterozygous pairs. So it's 2 to the second power, which equals 4. So that will give us the number of different gametes. Now, if the question asks, what are the different gametes? There is a little trick that you can use to help you figure this out. So this is my method to figure out the gametes. I make these tree diagrams. So when we have these chromosomes, right? A is on one chromosome, B is on another, C is on another, D is on another. They are going to assort independently. So when we form gametes, remember that big A, those are the homologous pairs, and big B is the homologous pair. So remember that when we go to form gametes, we are going to have independent assortment. So our gametes will always have one A, one B, one C, and one D. Your gamete should have one of each letter. So now we need to figure out what are those different possibilities. So big A, notice the only type of offspring that the children, or the only type of gamete that could be produced is a big A. That big A could pair, there's 2 choices, big B, or it could pair with little b. Now, the one with the big B could pair with big C or little c. Those are equal possibilities. The little b could pair with big C or little c. So we have to branch because there's 2 possibilities. And then the D's, notice the only option is that you're definitely going to give your offspring the big D. So I don't need to branch. I'm just going to go straight across. Big D, big D, big D, big D. There's no branch at that point because there aren't 2 options. There's only one option. So now you can just read them going from the top down. So if I were to write these out, big A, big B, big C, big D, and then I follow this one here, big A, big B, little c, big D. Then I have this one here. So I have to follow it along. So big A, little b, big C, big D. And the last one is going to be big A, little b, little c, big D. And so notice when I wrote them out, there were 4 different possible gametes, which matched my equation that I used at the top. So if you just need to know the number of different gametes, you can use the equation 2 to the n power, where n is the number of heterozygous pairs of alleles. So there were 2 heterozygous pairs, so it was 2 to the second power, which is 4. If you need to know what are the possible gametes, remember that you need to start with one of each letter. Your gamete has to have one A, it has to have one B, it has to have one C, and it has to have one D, because those are all homologous pairs, and the gametes are going to give one of those homologous pairs. Now, notice when we started this is big A. Had that been big A, little a, all you would do is underneath, so notice I started with big A here. If that had been heterozygous to start, you would just start with a second tree. So that would be little a. And then you would do the same thing. So let's say this had been big A, little a. Then that could pair with big B or little b. That could pair with big C or little c. Big C, little c. And then D, 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 D. It's always going to pair with a big D. So this type of diagram just makes sure that you hit all of the different combinations because you're writing it out for each possibility. And so this is how I solve a problem where it's asked for what are the possible gametes. I like to use these tree diagrams to make sure that I hit each one of those probabilities. So here is a practice problem. Again, this is going to be one that you're going to post your response on Canvas. An individual with the genotype big A, big B, big C, little c, big D, big E, little e, can produce how many different types of gametes, and what are the possible gametes? So you need to write out what are the possible gametes as well as how many different types of gametes are they. So again, go back and review the slide before if you are still unsure of how to solve this problem. Remember that whenever there's only one possibility, don't branch that, just draw a line straight across. So you're going to solve this problem. So here's practice problem number 4. I'm going to do this one with you. If a cross is made between a purebred green heterozygous smooth plant and a heterozygous yellow smooth plant, what is the probability that the offspring will be yellow and smooth? Now, in this particular question, I did give you the genotype, but in the other questions, I might not. So you need to be able to read the question and then figure out the genotype. So I start by writing down the phenotypes of the parents. So I wrote down the phenotypes of the parents. So green, heterozygous smooth, crossed with heterozygous yellow and smooth. Now, the next thing you're going to want to do is you're going to want to write the genotypes. Now, before I do that, I like to write what the possibility for genotypes would be. So let me show you what I mean. So I wrote myself a little key to help me keep track of what's what. So I wrote, well, big Y something is yellow. And what that little line means is that big Y can be yellow, or big Y can be yellow. So that just means that any second allele. Green has to be little y because it is recessive. Big S something is smooth. Little s is wrinkled. So in this case, we are crossing green, which we know has to be little y, crossed with heterozygous smooth. So little y, big S, little s. Heterozygous smooth tells you need a big S and a little s. And then the next parent is going to be heterozygous yellow smooth. So that's assuming it's heterozygous at both alleles. So there's heterozygous smooth, big Y, big S, little s. So there are 2 different ways you could approach this problem. For me, I like to treat them as independent crosses, meaning I like to cross the Y's separately and the S's separately. So that's what this is going to look like. So first I need to figure out what are the gametes for Y. So the green heterozygous smooth for the Y allele is the only option is the little y. So notice that on one side of the Punnett Square, we have the little y all by itself. I don't need to write both rows because it will come out the same. Heterozygous, there are 2 possible alleles, big Y or little y. So notice I put those on the outside too, big Y, little y. And then you're just going to fill in your Punnett Square. So my offspring have a choice of being big Y, or little y. Now I'm looking for the probability that they will be yellow. Yellow, remember, is big Y something. So what is the probability that they will be yellow? That is going to be 1/2. So 1/2 probability that they will be yellow. Now I need to do the crosses for the S's. So the green heterozygous smooth has 2 possible gametes for the S's. It could give a big S or it could give a little s. Again, be careful for letters that look alike. I tend to make my little s's very small relative to my big S so that I don't get them confused. And we're crossing that with another heterozygous smooth, which is big S, little s. So now I need to put my gametes on the outside of my Punnett Square and then fill in the result. So big S, little s on the outside of both sides of my Punnett Square, and then I fill it in. Big S, big S, big S, little s, big S, little s, little s, little s. So I'm looking for smooth. Remember that smooth is big S, any second allele. So notice that we have 3 out of 4. So notice that I am now going to have 3 out of 4 will be the S's. Now I just need to multiply my 2 probabilities. So 1/2 times 3/4. Multiply the numbers on the top, 3. Multiply the numbers on the bottom, 8. So my answer in this case is going to be 3/8. That is my probability of getting yellow and smooth. So 3/8 is my answer, and I did that by solving those crosses independently. Now, that's one way you could approach this problem. The other way to approach this would be to do the large Punnett Square. So in order to do the large Punnett Square, you will have to figure out the gametes for the two parents. So for green and smooth, notice it's little y, big S, little s. And so in the gametes, it should have one Y and one S. If your gametes don't have one of each letter, you didn't set it up properly. You have to have one of each letter. So again, I do my tree diagram. And so my little y is the only option for Y that can pair with a big S or a little s. So my two gametes for this parent are going to be little y, big S, little y, little s. Now I need to do that with the other parent. I'm going to change the color of ink just so it makes it easier to see the two parents, but I need to do the same thing for the other one. So I made my tree diagram, big Y, big S. That's one possible gamete. Big Y, little s. That is another possible gamete. Little y, big S. That's a possible gamete. And then little y, little s. And so now I need to set up my Punnett Square with the blue ones on one side of my Punnett Square and the red ones on the other. So this is how I set up my Punnett Square. The blue is on one side. There were 4 possible gametes, so I put all 4 on the outside. For the one that's red, there were 2 possibilities, little y, big S, or little y, little s. And so now all you're going to do is you're going to fill in your Punnett Square. So I put both the y's back together and both of the s's back together so that it makes it easier to see the genotypes. So I filled in my Punnett Square, I put my y's together and my s's together, and now I need to look for the probability that they are yellow and smooth. And so notice that for yellow, it's big Y, any second allele, and smooth, it's big S, any second allele. And so let's look on our Punnett Square and find them. And so we have 3 out of 8. So notice this one doesn't work because it's little s, little s. These all don't work because they're little y, little y. So notice that we got the same answer either way. So you could either treat them as independent crosses and multiply the probabilities like I showed you, or if you feel more comfortable doing the larger Punnett Square, you can do that as well. The one thing I'll say that students often struggle with doing the larger Punnett Square is that they forget to put a Y and an S together. Remember, the outside of your Punnett Square has to have both letters because those are homologous pairs, which means that Y is only going to give 1, and that will also be with an S, which has to give 1. So you have to make sure that these gametes on the outside have both letters. If they don't, it doesn't work. So those are your 2 options in how to solve this problem. So I have a question for you to practice with. Again, this is going to be one that you will post your answer on Canvas. In pea plants, yellow seeds are dominant over green seeds, and smooth seeds are dominant over wrinkled seeds. If a cross is made between a heterozygous yellow, wrinkled seed plant and a heterozygous yellow, smooth plant, what is the probability that the offspring will be yellow and wrinkled? And again, for the heterozygous yellow smooth, it's heterozygous at both. So what is the probability that the offspring will be yellow and wrinkled? And so you want to put your answer on Canvas. And once the due date closes, I will post the answers and how to solve those for you. So here are some examples of traits in humans and showing whether they're dominant or recessive. So freckles is dominant over no freckles. So no freckles would be recessive. Widow's peak, where your hair comes to a point, your hairline comes to a point, would be dominant over a straight hairline, where your hair goes straight across. Free earlobes, so your earlobe hangs down, that is dominant over attached earlobes. And so these are just some examples in humans. So when we look at human genetics, we often use a pedigree analysis in order to follow traits through generations. And so this is used when mating behavior can't be controlled, because obviously you can't tell people you need to mate with you. That would be unethical. So we use pedigrees to track traits within a family. And so this is used to infer probable genotypes from a family history. So if you see a circle represents a female. If it is open, meaning not shaded in, that is a normal female. If it's a closed circle, meaning it's shaded in, that is an affected female, meaning she has whatever trait it is that you're looking at. Open square is going to be male, so squares are male. Closed squares are going to be affected males. Diamonds would be sex undetermined. So like if somebody was pregnant with a child and they didn't know the gender of the child, it would be indicated temporarily as a diamond. When we see lines between circles and squares, that indicates a mating pair, meaning that those two people are going to have children, and then you'll see a line coming down and you'll see their children. So if you're looking at a dominant pedigree, every affected child must have an affected parent, always. Because if you have a child that has the trait and the trait is dominant, that means that she had to get that dominant allele from one of her parents, right? And so in a dominant pedigree, one of the things that you can look for when determining dominant or recessive is if you see a child that has a trait and neither parent does, that means that the trait has to be recessive. But in this case, we have a child that has it. Every affected child has an affected parent, and you can see that in this case. All non-affected individuals must be homozygous recessive. So notice that for all of these that are not filled in, we know that the genotype has to be little w. So this is comparing widow's peak versus non-widow's peak, right? And so this one is little w. You can always start off, the way I start off when I'm trying to solve a pedigree is I start with what we know right away. So all of the ones that are not filled in, I know need 2 lowercase letters. Then I start with the ones that are filled in and I write big W blank. So when I write big W a line, that means that until I look at the offspring, I can't say what the second allele is. However, notice that this pair at the top right here, that pair has a child that is little w. If the child is little w, that means that the little w was given by both parents. And if both parents gave a little w, that means that the filled in circle has to have the second allele to be little w. And so we know that the second allele is the little w. Now, if we look at these two parents, notice they have a child that is little w, which means the second allele for both of these parents has to be a little w. Same thing here. If we look here, this second allele we know has to be little w because we have children that are little w. This one, right? This one doesn't have children. We can't use information about their children, but we can use information from the parent. They have a parent that is little w, which means that this parent can only give a little w, which tells you that this allele has to be a little w. In the case of this one here, we can't determine, there's not enough information to determine the second allele. Because if these parents are big W, little w, by chance the child could be big W, or big W, little w. We don't know. We can't make an assumption on that because we don't know what the second allele is going to be. And so this one is one that's unknown. So what are the criteria for an autosomal dominant trait? Males and females can be affected. Male-to-male transmission can occur. That is going to be different than if we look at sex-linked traits, which we'll talk about later. But male-to-male transmission can occur. Males and females transmit the trait with equal frequency, which again tells us that it's autosomal. Remember that autosomes are chromosomes that don't determine gender. Successive generations are affected, so you see this in multiple generations. And then the transmission stops after a generation in which no one inherits the mutation, right? So if nobody inherits the mutation, that trait no longer is passed on. So a recessive pedigree. In a recessive pedigree, every affected individual must be homozygous recessive. So notice that all of the ones that are filled in are little f, little f. That's attached earlobes. That's recessive. Affected children may or may not have an affected parent. So notice if we look down here, right, here are two parents, neither of them have the trait, and they have offspring that have it. That right away, when you see that, you go, aha, this trait has to be recessive. Because remember, if this trait is dominant, right, if this was a dominant trait, that means that the dominant allele is here, which means one of the two parents had to give the dominant allele. But we don't see that. So if you see this type of transmission where neither parent has it, but they have a child that does, that tells you that that is a recessive pedigree. Now, the people who don't have it, they are heterozygous, right? In this case, when they have a child that has it, they are heterozygous, and we call them carriers. They carry the allele, but they don't have it. And so they could, by chance, give that recessive allele to their offspring. And so in a recessive pedigree, you can start by filling in all of the filled in ones are little f, little f. All of the not filled in are big F something. So again, you would draw that line and then use the information in the table to figure out the second allele. So the reason we know this one is a little f is because they have offspring that are little f, which means a little f came from both parents. So we know those have to be little f, or big F, little f. In this case, this child could be either. We can't say. So this is still an unknown. It could be big F, or big F, little f. This one we know because the child has it. So the second allele has to be a little f. This one we know has to be a carrier because the child has it and is little f, little f. And so there are certain things that you can look for that help you when determining whether a pedigree is going to be a dominant trait or a recessive trait. So let's look at some examples of traits in humans. So there are a few inherited diseases that are caused by dominant alleles. Some examples of dominant alleles would be achondroplasia, which is a form of dwarfism, Huntington's disease, which is a neurological disease. The gene for achondroplasia was discovered by Professor John Wasmuth at UCI. Professor Leslie Thompson at UCI works on these diseases. So these are worked at locally, but these are dominant alleles. Now, if you're talking about a dominant trait, a dominant allele can't be a disease that is going to be lethal early on. And the reason for that is if the trait is lethal early on, before people have kids, that trait will never be passed on and that'll be wiped out from that population. These conditions either are not lethal or they come on late. Huntington's disease, for example, is a neurological disease. It has a late onset, often in the 40s or 50s. And so by then, people have already had children and they've already passed that trait on to their children because they didn't know that they had it. And so some examples of things that can happen. So we can have dwarf parents crossed with normal height people. When you see somebody who has achondroplasia, they can't be big D, big D. Big D, if someone were to inherit two dominant alleles, it's lethal. It doesn't happen. So if you see somebody with achondroplasia, they have to be big D, little d. So we have one parent with achondroplasia, they're big D, little d, crossed with a normal height parent. So what we're looking at is the sperm could either have big D or little d. The egg could have little d, little d. And so if we fill in our Punnett Square, notice that if you have somebody with achondroplasia with a kid with normal height or with another parent with normal height, the probability of having a child with achondroplasia is 1/2. The probability of having a child with normal height, 1/2. Now, if you've ever seen that show, Little People, Big World, right? They have dwarfism and they have height that are normal height children. Now, I don't know whether they have achondroplasia or some other form, but what I can say is notice that you have 2 parents that have dwarfism and you have children that do not. That's because those 2 parents with achondroplasia can carry the normal allele. That can give rise to a normal height child. And so you can have instances where the children don't have the condition. So if you're curious about any traits and how they're inherited, there is this online Mendelian Inheritance in Man, this website through the government, that allows you to check into different diseases and find out how they're inherited. So things that we know. So what chromosome is that gene linked on? How is it inherited, etc. And so that's just kind of a cool little website if you want to learn more about a particular disease. So in an autosomal dominant trait does not skip generations. Again, you can't have it skip a generation because if the child has the disease, the parents, at least one parent has to have it as well. They can affect both sexes, so both males and females can have the trait. That's what tells you autosomal. Again, you'll see in a little bit how sex-linked is going to be different. And so what you're looking at in this table is something called polydectaly, which is extra fingers or extra toes. And so this is a pedigree of a family showing how it is transmitted. And so you'll notice that for every filled in one that you see, a square or a circle, remember that the squares are going to be males, the circles are going to be females. Notice it affects both males and females. So there's no correlation with who's infected more, males or females. It infects both. So this tells you autosomal dominant. More diseases, however, are caused by recessive alleles. And that's because if somebody is a carrier of a trait, you can have 2 parents that don't have the disease and then a child that does. So some examples of recessive allele traits or diseases, albinism, so lack of pigmentation, sickle cell disease, so sickle cell anemia, cystic fibrosis. Cystic fibrosis has patients who are born with this have defective chloride channels. And as a result, because they don't move salt properly, they also don't move water properly. And so patients with CF have these thick secretions in their lungs. Oftentimes they might need a heart-lung transplant. They have problems with their pancreas. There's a whole host of problems that come out with cystic fibrosis. Tay Sachs disease, we talked about this in the lecture about organelles. Tay Sachs, remember, is a lysosomal disorder in which patients who have inherited this disease have a mutation in their enzyme that helps metabolize lipids in the brain, for example. And so what ends up happening is that people who are born with Tay Sachs can't metabolize lipids, and the lipids build up and they accumulate in the brain, and the children become deaf, blind, unable to swallow, and typically die by age 4. And so Tay Sachs is a recessive allele. You have 2 parents that don't have Tay Sachs and may not even know that they carry it, and then they have children that have it. And then PKU. PKU is another one that I talked about, phenylketonuria. In phenylketonuria, patients who are born with this don't metabolize phenylalanine. And so for them, phenylalanine is an amino acid, and it builds up to toxic levels when the enzyme is missing, and it causes damage to the brain. And especially in children while their brain is developing, that brain damage can lead to major impairments. So those are recessive alleles. So notice that we can have 2 parents, big A, little a, big A, little a, and neither parent has the trait. In this case, we're looking at albinism. And so we get big A, little a. Those are the 2 gametes. Big A, little a. Those are the 2 gametes. And if we look at the probability of getting a child who is albino, we have 1/4, right? There's a 1 out of 4 chance that the child will have albinism. Now, does that mean that that is what's going to happen? Answer is no. Remember that people have very small sample sizes. They don't have a lot of children, and often the ratios don't work out the way you would expect. An example of this is my kid's dad, so my ex. He does not have PKU, but both of his brothers do. Neither of his parents have PKU, so his parents were just like this. They were carriers. They had no idea that they carried PKU. Nobody else in their family had ever had it. They were carriers, and 2 carriers happened to come together, and they had 3 boys. Now, you would expect that 1 out of 4 would end up with PKU, right? There's a 1 in 4 chance. Now, that's what you expect. They only had 3 boys, and they got 2 out of 3. So their probability worked out to be a lot higher than what you would expect. They got 2/3 of their children had PKU, and 1/3 does not. Now, in terms of when you're solving genetic problems, you have to be very careful of the way that the question is worded. So if I gave you a question and I said, there are 2 parents who do not have PKU. They have a child that has PKU. So what that tells you is that both parents are carriers. So they have a child that has PKU. If it said, what is the probability that they will have a normal child, right? Will have, they haven't had it yet. The probability of having a normal child would be 3 out of 4. Now, watch if I asked it a slightly different way. So 2 parents do not have PKU, but they do have a child that has PKU. Again, that tells you that they are both carriers. And then I say they have a child, so like my ex, they have a son who does not have PKU. So they have a son who does not have PKU. What is the probability that the son is a carrier of PKU? So if you look at this, right, out of all the possibilities, 2 out of 4 would be a carrier. But notice that's not the way the question was asked. The question was asked, they have a child who does not have PKU. Automatically, that gets rid of the little a possibility. The child is already born. They don't have PKU. So the little a, the PKU option is out. If they said, will have a child that is a carrier, that would be different. But the couple has a child. The child does not have PKU. What is the probability that the child is a carrier for PKU? The answer is 2/3. Because when we get rid of the little a option, meaning the PKU option, there are only 3 possibilities left. Normal, not a carrier, or normal and carrier. Notice if you look at the pedigree, the Punnett Square, you'll notice that 2 out of the 3 are carriers if it's normal. And so again, you have to watch the way that the questions are worded in order to get the right answer. So for autosomal recessive traits, things that you're looking for, males and females can be affected. So again, it doesn't favor one gender or the other. Affected males and females can transmit the gene unless it causes death before reproductive age, right? And so, for example, Tay Sachs, if somebody is born with Tay Sachs, they themselves are not going to pass it on because they will die before reproductive age. But carriers can keep that trait in a population. Traits can skip generations, again, because you could have children that are carriers but don't have the disease. Parents of an infected individual are always either heterozygous or have the trait, right? Because if the individual is infected, that means that they had to get the recessive allele from both parents. So at least one parent, the one parent can be heterozygous or has the disease as well. Conditions likely to occur in families when we have marriage between relatives. So think of incest. This is often kept in that population more when you have breeding within a family because it keeps that trait in that population. So, question for you. PKU is a disease caused by a recessive allele. A man who has PKU marries a woman who does not have PKU. The woman's father has PKU. What is the probability that the couple will have a child with PKU? So I will fill this in as a pedigree. So this is our pedigree. The man has PKU, so he's filled in, marries a woman who does not have PKU, so she is not filled in. But the woman's father, notice going up from the woman, her father has PKU. I didn't fill in the mother because we don't need that piece of information to fill this in. So we want to start with first filling in the PKU. PKU is recessive. So what alleles do people who have PKU have to have? Let's say we use the letter A. Well, anybody who has PKU has to be little a. So I start by filling that in. So the ones that have been filled in are little a. The woman who does not have PKU, we know right away just from that first part that she is big A something. Now notice her dad has PKU. What does her second allele have to be? And the answer is her second allele has to be a little a because that's all dad can give. He has to give a little a. That's his only choice. So we know that she is big A, little a. So she is big A, little a. The husband is little a. So if we fill out a Punnett Square for those two people, we have big A, little a. That's from mom. Dad has the little a. And so now all we do is we fill in our Punnett Square. And so when we fill this in, we have a 1/2 probability of having PKU. The little a is PKU. So 1/2 probability of having PKU. And so this is how you would go about solving this type of problem. So here are some examples of inherited disorders. You do not need to memorize this table. It was just to give you an idea of some autosomal dominant and recessive disorders. So we'll start with albinism. The major symptom of that would be lack of pigments in hair, skin, and eyes. The incidence of this, it's very rare, 1 in 22,000. So 1 in 22,000. Cystic fibrosis, symptom, excess mucus in lungs, digestive tract, liver, increased susceptibility to infections, death in early childhood unless treated and managed, 1 in 1,800. And it's higher prevalence in European Americans. And so one of the things when you go to have kids, depending on your ancestry, you can be asked if you want to be tested to see if you are carriers of certain diseases. So one of the ones that I decided to get done was to see if I was a carrier of cystic fibrosis, because based on my ancestry, that would be a disease that I would be more likely to carry. So I got tested. I am not a carrier. So my kid's dad did not need to get tested because if I'm not a carrier, I can't have a kid with cystic fibrosis. And so depending on your background, right, if you are from African descent, you might then be tested to see if you're a carrier of sickle cell disease. So depending on your background, that might change which ones you might want to be screened, or you might not want to know. That's completely a personal choice. So cystic fibrosis, then we have PKU, phenylketonuria. This is going to lead to an accumulation of phenylalanine in the blood, lack of normal skin pigment. So people who have this are typically more fair-skinned because the phenylalanine is used in the production of melanin. Mental retardation unless treated. So again, you would just treat this by managing with a low protein diet. As long as it's managed with a low diet, no brain damage occurs. It's when protein levels get too high that it becomes a problem. Notice the incidence of this. It is extremely rare. 1 in 100,000. 1 in 100,000. And the chance of my ex's 2 brothers from having it is pretty much unheard of, because again, the probability of having 1 kid with it is 1 out of 4, and the probability of having 2, right, is pretty low. So they just happen to get very unlucky. Sickle cell disease causes the sickled red blood cells and damage to many tissues. That has an incidence of about 1 in 500 people of African American descent. Interestingly, for sickle cell disease, what makes sickle cell disease so interesting is that there's a reason that it's more prevalent in people of African descent. And that is that a major problem in Africa is malaria. And so malaria is transmitted by the bite of a female Anopheles mosquito, a special type of mosquito. So in Africa, where malaria is very problematic, actually carrying the sickle cell allele, meaning you don't have sickle cell disease, but you carry it. If you carry the sickle cell allele under certain conditions, it can cause the phenotype to manifest somewhat, meaning that you're not going to have as many sickled red blood cells, but you may have some. So people who are carriers for sickle cell allele, they are actually protected against malaria. So in Africa, it's actually a good thing to be a carrier for sickle cell disease because it protects against malaria. And so it's actually been selected for in that population. People who are carriers are more likely to survive than those that are not carriers. And so that is why that trait, that particular trait, is more prevalent in that group. Tay Sachs leads to a lipid accumulation in brain cells because, again, it's a deficiency in a lysosome enzyme that's used to metabolize lipids. It causes mental deficiency, blindness, and death in early childhood. 1 in every 3,500 people of European Jewish descent. So if you fall in that category, you might be asked if you would like to be screened for Tay Sachs. Then we get to our dominant disorders. Achondroplasia. Achondroplasia, dwarfism. 1 in 25,000. Alzheimer's disease, there's one type. The incidence of this is not really known, but Alzheimer's has mental deterioration, usually strikes late in life. Huntington's disease. So Huntington's disease, mental deterioration and uncontrolled movements. Again, it's a neurodegenerative disorder. It comes on later in life. Has an incidence of about 1 in 25,000. And then we have hypercholestemia, which leads to excess cholesterol in the blood and eventually leads to heart disease. 1 in about every 500 people. So that one is not quite as rare. And actually, hypercholestemia, while it's listed here as dominant, it actually is not fully dominant. It is what we call incomplete dominance. And we'll talk about that more in just a minute. So there are ways to test a fetus to see if they have that disorder. So for example, let's say I was Eastern European Jewish descent, right? And I got screened for Tay Sachs. And it was discovered that I'm a carrier. And my partner also got tested and he found out he's a carrier. And we wanted to know then if we were both carriers, then there would be a probability that the kid might have Tay Sachs and would die at an early age. And so there are ways to test a fetus for genetic disorders. The most common and the kind of historical ways would be through one of 2 methods. One is referred to as amniocentesis, and the other one is chorionic villus sampling, which is abbreviated CVS. Think of like the drugstore. Amniocentesis is where they put a large needle and they put it into the uterus and they draw out some of the amniotic fluid. Now, the amniotic fluid is going to contain cells from the fetus. And so the amniotic fluid is going to be drawn out. It's going to contain fetal cells. And they can then take those fetal cells, they can run biochemical tests, they can culture the cells and do a karyotype. We talked about the karyotype in the meiosis chapter, right? Looking at chromosomes. So let's say they were looking to see if the fetus had Down syndrome, right? A karyotype would be useful because you're looking for an extra copy of chromosome 21. Now, for amniocentesis, this is typically done around approximately 16 weeks of pregnancy. So this is a little bit later on in pregnancy compared to CVS sampling. The chorionic villus sampling can be done earlier at about 12 weeks. And in that method, what they're doing is they put a tube through the cervix, and it's a little suction tube, and they suck a little bit of this chorionic villi, which is derived from the placenta. And this will contain fetal cells. And again, they can do biochemical tests or karyotyping. Now, in CVS, it's an advantage in that it's much faster. It can be done earlier on in the pregnancy to see if there's a problem earlier on, etc. Now, the reason they don't routinely do these tests is that these both have a risk of miscarriage. You might have an increased risk of miscarriage by going through these procedures. And so typically they're not going to do these procedures unless you are high risk for one reason or another, or you're a carrier of a disease, etc. And so these are some ways that they can test a fetus for particular genetic disorders. So here is a practice problem. So after I read this, I would like you to pause and attempt to do this on your own. So the pedigree below traces the inheritance of alkaptonuria, a biochemical disorder. Affected individuals, indicated here by colored circles and squares, are unable to break down a substance called alkapton, which colors the urines and stains body tissues. Does alkaptonuria appear to be caused by a dominant allele or recessive allele? And then fill in the genotypes of the individuals whose genotypes can be deduced. What are the possible for each of the other individuals? So first you want to look at this collectively and see if you can identify if it's a dominant or a recessive. There is one thing that is a sure giveaway, what type of inheritance it is. And so you want to see if you can find that. And then once you figure out if it's dominant or recessive, you want to fill in the possible genotypes. It's like a puzzle. You need to figure out what are the two alleles, or in some cases, there might be more than one possibility, and you want to write those possibilities out. So take a minute, pause, and when you're ready, push play to hear the answer. So how is this trait inherited? The answer is that this is recessive. Notice we have Anne and we have Michael, neither of which has the disease, but they have a daughter, Carla, who has it. So that tells you that that cannot be a dominant trait, because if Carla had it and this is dominant, that would mean that Carla has a big A, which means that would have came from one of her parents, but neither parent has it. So that right there, this family here, tells you without a doubt that this trait is inherited by a recessive allele. So now what we can do is we can fill in the table. Everybody who is affected, if this is recessive, is going to be little a, little a. And I start by the people that are unaffected. They are big A something. So I'm just going to start by putting that on here first, and then we'll fill in the second allele for the dominant trait. Okay, so all of the ones that are filled in were given little a, little a. All of the ones, sorry, George should have a big A line also, but all of the ones that are not filled in are big A something. All right, so let's figure out the pieces of the puzzle. All right, first of all, let's start with George. George, we know, is big A something. We need to figure out his second allele, right? If it's a dominant, if George is dominant, we know he's big A something. Could be big A, could be big A, little a. But what about his children tells you what his second allele is? The answer is that he has children that are little a, which means that both parents gave a little a. So George's second allele has to be little a, so that we know. What about Sam and Anne? So Sam and Anne are children of George and Arlene. Do we know their second allele? Answer, yes, we know, because Arlene is little a, which means that Sam and Anne both have to have little a. Now, if we look at Anne and Michael, do we know Michael's second allele? Yes, because Carla, their daughter, is little a, which means that she got a little a from both parents. Now let's make our way over to Sandra and Tom. So let's first start with Daniel and Alan. Daniel and Alan are kids of Sandra and Tom. Do we know Daniel and Alan's second allele? Answer is yes, because look at their dad, Tom. Their dad, Tom, is little a, which means that the dad always gives a little a. So we know that Daniel and Alan, their second allele has to be a little a. Now, we know their second allele has to be a little a. If we look at Sandra, can we determine Sandra's information based on what's here? The answer is no. Yes, if Sandra was big A, little a, we would expect that half of them would be little a, and we don't see that. But with a sample size of 2, meaning they only have 2 kids, you might not ever see that. So you cannot say with certainty that Sandra is big A, little a, or big A. Sandra, we don't know the second allele. She could be homozygous dominant, or she could be heterozygous. Alan and Tina. So for Tina, do we know Tina's second allele? Nope. Do we know Christopher's second allele? No. There's not enough information there to allow us to determine that. So Sandra, Tina, and Christopher are all unknown. We know that they're big A something. They could be big A, or big A, little a. There's not enough information there to tell you what they are. So, question for you. Astigmatism is dominant over normal vision. What is the probability of offspring having astigmatism if both parents have astigmatism? Both parents each had a parent with normal vision. So, red, 1, yellow, 1/2, green, 1/4, blue, 3/4, or purple, none. So pause your video, and when you're ready, push play. All right, so here is our pedigree. So we have 2 parents that both have astigmatism, and they each have a parent with normal vision. So notice that the parent is drawn, not filled in. So now we need to figure out what are the genotypes. So notice that if this is a dominant trait, we know that the filled in ones are big A something. So they're big A something. Now, they each have a parent with normal vision, which means that the parent with normal vision has to be little a. So if they each have a parent with normal vision, what does their second allele have to be? Answer is second allele has to be a little a, because that one parent can only give a little a. So we know that they are both big A, little a, and then we fill in our Punnett Square. So our cross is between big A, little a, big A, little a. So we fill in our Punnett Square, big A, little a, fill it in. What is the probability of the offspring having astigmatism if both parents have astigmatism? So notice that astigmatism is dominant, so it's any of the ones with a big A. So the answer is blue, 3/4. 3/4 will have astigmatism because they have the dominant allele. And so that's the answer to that question. So now we're going to move on and talk about variations from classic Mendelian genetics, because not everything has just 2 alleles and everything is just dominant or recessive. It's a lot more complex than that. So this I just put here to summarize the different variations. I'm going to walk through each of these examples, so I'm not going to cover them here, but I did want it to be all on one slide so that you have that information. So we're going to start with our incomplete dominance. Incomplete dominance is where the heterozygous have a phenotype in between the dominant and the recessive. So if you remember back to Mendel's experiments, when he crossed purple and white, the offspring were either purple or white. There was no lavender. There was no color in between. They were either the dominant phenotype or the recessive. There was no in between. However, if we look at carnations, carnations have incomplete dominance. If you cross a red with a white, The red with the white is going to give pink. That's a phenotype in between. So that heterozygous has one red allele and one white allele. Now, you'll notice that we have a different type of notation here, and that is that we don't want to use an uppercase and a lowercase because that indicates that a gene is dominant and one is recessive. That is not the case here. So notice that in this case, we have the C gene, and on the C gene, we can have an allele. So the allele could be the red allele or the white allele. That's why you have the superscript R or W. So the red is CR, 2 red alleles. The white is CW. When they have offspring, the offspring are CR and CW. They have one red allele, one white allele. They make half as much pigment. They're pink. If we cross 2 of those F1 generations, if we cross 2 of the pink flowers, right, the gametes that are possible are the CR or the CW. So I have to put that on my Punnett Square. So if you notice on the outside of the Punnett Square, you see CR, CW on both sides. Then you fill it in. So this one is CR, that would be red. This one is CR, CW, that would be pink. This one is CR, CW, that is pink because it has one red and one white. And this one here underneath there says CW, CW, 2 white alleles, which makes it white. And so in incomplete dominance, you'll notice that normally when you cross the heterozygous for the pea plants, right, the purple and the white, and when you cross those heterozygous, you get a ratio of 3/4 purple to 1/4 white. When we look at incomplete dominance, we have a different setup. We have a 1-2-1 setup. 1 red, 2 pink, 1 white. The probabilities are different, which shows the incomplete dominance. And so this would be what you would get in this example. So let's talk about why it works this way. So again, the C gene encodes a protein that catalyzes the production of red pigment, meaning it allows the cell to make red pigment. The CR encodes a functional version of the enzyme, meaning it will make the red pigment. CW encodes a broken version of the enzyme. It's mutated and the enzyme doesn't work, so it does not produce red pigment. For red color, plants need 2 doses of the functional enzyme. With 1 dose, not enough enzyme is made to produce enough pigments. And so they don't produce enough pigments to be red, and instead it results in pink flowers. They make some pigment, but not enough to make them red. So if we're looking CW, right, 0 doses of functional pigment, 2 doses of the broken pigment, which is why they're white. They're the absence of color because they don't have any functional red pigment. CRCW, the heterozygous, has 1 dose of functional, that's the red, 1 dose of the broken, that's the white. So they produce half as much pigment. They're not red, they're white, or they're pink. If we look at CRCR, that is 2 doses of functional enzyme, 0 doses of the broken enzyme, and as a result, it produces enough pigment to be red flowers. So an example in humans of incomplete dominance is familial hypercholestemia. You might recall that we talked about this back in our transport lecture about LDL receptors, and that LDL receptors bind to LDL. LDL is your cholesterol carriers in the bloodstream. And so the LDL receptors bind to the LDL, and they take the LDL into the cell, which keeps blood cholesterol levels low. Now, if somebody is heterozygous, now again, this type of notation that's written here is not correct because it's not dominant and recessive. It actually is incomplete dominance, but we'll overlook that for this. But let's say this is just talking about it being heterozygous. right? They have one functional gene and one mutated gene. So what that means is in terms of the LDL receptors, they're going to make half as much functional LDL receptors because there's only one allele to make LDL receptors. The other one's broken. So if they're heterozygous, they have a more mild form of hypercholestemia, meaning their cholesterol levels are not quite as high. If they are homozygous for the inability to make LDL receptors, that means they have 0 copies of functional receptors. Without the LDL receptors, cholesterol can't get into the cells and instead stays in the bloodstream, and that is going to lead to very high cholesterol. Usually patients who have this might end up having a cardiac episode by the time they're in their 20s. This is extremely high cholesterol. So what you're looking at is this is showing cholesterol levels for different types of populations. So the general population, plasma cholesterol levels, you want your cholesterol to be under 200. So this is kind of a range of what cholesterol levels might look like. For people who have genetically high cholesterol, if they're heterozygous, their levels might be in the 400s, 500s range. I am one of these people. No matter how well I eat or exercise, my cholesterol will still be high because I don't have functional LDL receptors. For patients who are homozygous for familial hypercholestemia, they inherit 2 bad copies. They don't make the LDL receptors. And so notice that their cholesterol levels are up to 1,000. So 5 times higher than what they normally should be. And so what ends up happening is if we look at these blood vessels, this is a plaque, this is this buildup forming that is going to impede blood flow. And that can lead to a heart attack, for example. And again, if they are homozygous for familial hypercholestemia, oftentimes these patients will have heart attacks by their 20s because their cholesterol levels are just that high. So question for you. If a cross is made between a red flower plant and a pink flower plant, what is the probability that the plant will have white flowers? Red, 0, yellow, 25, green, 50, blue, 75, purple, 100. So push pause, work through this, and when you're ready, push play. So if you said red, 0%, you would be correct. So let's write this out. So the red plant is going to be CR, right? You have to have 2 red alleles to be red. Pink is going to be CR and CW, half as much pigment, which is why they are pink. So now we need to make our Punnett Square. So if I make my Punnett Square, the pink plant has 2 possible gametes, CR or CW. The red only has 1 possible gamete, CR. So if I fill this in, one offspring is going to be CR, the other one is going to be CR, CW. So we have a 1/2 probability of getting red flowers, a 1/2 probability of getting pink flowers, but there's no chance of getting white flowers because white flowers would need to inherit 2 CWs. Notice that the red has no CW to give, therefore white flowers are not a possibility. So we're going to continue on with our differences in Mendelian genetics. And so the next one that we're going to talk about is both codominance and multiple alleles. Multiple alleles is when there are more than 2 alleles found in the population. So again, if we think about Mendelian's genetics, he had purple flowers, white flowers, right? There were 2 alleles possible. In the case of multiple alleles, there might be more than 2 alleles. However, an individual can only carry 2 of those alleles because the individual is going to be diploid and will have 2 of the homologous pairs. So even though more than 2 alleles exist within the population, a diploid individual can only carry 2 of those alleles. An example of multiple alleles is the ABO blood groups. There are 3 alleles, and these 3 alleles lead to 4 phenotypes. So there's the A allele, the B allele, and the O allele. So 3 different alleles possible. But those 3 alleles can give us 4 phenotypes, 4 different blood types: type A, type B, type AB, or type O. Codominance means that neither allele is dominant over the other. Expression of both alleles is observed as a distinct phenotype in the heterozygous. That is different than incomplete. Incomplete has a phenotype in between. Codominance, you actually see both. Both characteristics are actually seen. Codominance is seen for AB blood. If somebody has AB blood, they have the A allele and they have the B allele. On their red blood cells, they make both the A carbohydrate and the B. Both alleles are being seen. And so let's take a look at this. So if we look at blood type, remember I said that there are 3 possible alleles. The O allele is written as a little I because the O allele is going to be recessive. The A allele is written as a capital I because that allele is dominant over O. So it's a capital I and then an A letter next to it. That's the A allele. B allele is written as I, capital I, with a B next to it. Now, the way that it's written in this notation is that the A and the B are dominant over O, which is why they're given a capital I, but the A and the B are codominant. So that's why they're both written with a capital. Because if you look here, if you inherit an A and a B allele, your blood type is going to be AB. You actually see both. So what do I mean by you see both? Well, the A allele is going to encode for an A-type carbohydrate on the surface of the red blood cells. So people who are blood type A produce this A carbohydrate. People who are blood type B produce a B carbohydrate on the surface of the red blood cells. So this person that is B produces this B carbohydrate. People who are AB make both the A and the B. They are both being expressed. That's different again than incomplete where it's in between. This is not in between. This is you're seeing both. So let's walk through it. O allele is recessive. So the only way that you can be blood type O is to be little I, little I. Little I is going to give O blood type. This is recessive, and what that basically means is that the red blood cells, think of it like the red blood cells are naked. They don't make an A carbohydrate. They don't make a B carbohydrate. They don't make either. So somebody who is blood type O, they can receive O blood. So they can receive O blood because O is seen as self, right? Because other O blood does not have those carbohydrates on it. However, if you're blood type O, you cannot receive A blood. Because if you're O, your blood is going to make anti-A antibodies. It will see that A carbohydrate as being foreign because their blood cells don't have it. Their blood will also make anti-B antibodies because, again, there's no B carbohydrate on the surface of that red blood cell, and B is seen as foreign. So what happens is when you're testing to see which blood types that can be given to a particular person, notice O is okay. A causes this agglutination. So if somebody with blood type O was given A blood, the person with O has anti-A antibodies because it sees A as being foreign. When the person with O has anti-A antibodies, if given A blood, it causes the blood to do this agglutination or this clumping. People who are type O cannot receive B blood because, again, B is seen as foreign because somebody who's O makes anti-B antibodies. And somebody who's O cannot receive AB because both A and B are seen as foreign. Now, in terms of blood type A has 2 possible genotypes. The person could be IA, meaning they inherit 2 A alleles, or the person could be IA little I. So they have an A allele and an O allele. Remember that the O allele is recessive. It's not seen. So somebody who has this blood here for type A, they make A carbohydrates. Now, the person who is A is not going to make anti-A antibodies because A is seen as self. However, they will make anti-B antibodies because B is seen as foreign. Somebody who's A does not make B carbohydrates. So somebody who is blood type A can receive O because O blood is naked. There's nothing for their blood to react against. They can receive A, but they can't receive anything with a B. So they can't get B and they can't get AB. Somebody who is blood type B, IB, or IB little I. So they could have two B alleles or a B allele and an O allele because again, the O is recessive. They make a B carbohydrate. So they don't make anti-B antibodies because B is seen as self. But somebody who's blood type B has anti-A antibodies because A is seen as foreign. So what that means is if somebody is blood type B, they can receive blood from O. O is, right, naked. There's nothing seen as foreign. They can receive B, but they can't receive anything with an A. So no blood type A, and they can't receive AB. If somebody is blood type AB, A and B, remember, are codominant. So the only way you get somebody who is blood type AB is that they have an A allele and a B allele. They make both carbohydrates, both the A and the B. So they don't make anti-A antibodies and they don't make anti-B antibodies because those carbohydrates are seen as self. They're not foreign, they're self. So somebody who is AB can receive blood from O. O is naked. They can receive blood from A because A is seen as self. They can receive B and they can receive AB. So notice something interesting. Which one is the universal donor, meaning that they can give blood to anybody? The answer is O. O is your universal donor. So if you ever watch medical shows and you see the people working in the ER, you might hear them say, we need O neg stat. So that's O blood. Negative is referring to the RH factor, which we won't really have time to talk about. And stat just means fast. So when they're in a trauma situation and they don't have time to determine blood type, to be safe, they'll give the person O negative blood so that anybody can receive that. AB is what we call the universal recipient. They can receive blood from anybody, right? They can receive blood from anybody. Now, I am blood type B. So I have two possible genotypes. What pieces of information might help me to figure out my second allele? Answer is either look at my parents or my children. Either one could give me an idea of what my genotype would be. Because let's say I'm blood type B, that's what I am, I'm B. Let's say I found out that one of my kids is O. So let's say my son is O. If my son is O, he has to be little I, which means what does my second allele have to be? My second allele has to be that O allele, that little I, because I have a kid who is little I, which means he got a little I from me. So you can use information in family trees to try and figure out what the second allele is. But even then, that's not always possible. So like if my parents were AB and B, I don't know my second allele, right? I could be either possibility. Now, let's think about what some non-possibilities would be. So one time I asked my grandma what her blood type was. And my mom said she's AB. And my grandma, her mom, my grandma claimed that she was O. So my grandma said she's O and my mom is AB. Is that even possible? Answer is no, that is not possible, right? Because if my grandma was O, she would be little I. There would be no way that my mom could be AB unless that really isn't her mom. So that's not possible. Turns out my grandma was incorrect. She is blood type B. Problem solved. Not an issue anymore. But when she was really adamant that she was O, I knew something was a little off there. There's no way my grandma is O if my mom is AB. Now, what if it was the other way? What if my mom was O? Could her mom be AB? Could my grandma be AB and my mom be O? No, that's not possible either. Because if my grandma was the AB, my grandma would be this. And that would mean that my mom would be O, which would be this. This O child could not have a parent that is AB because they had to get a little I from one of the parents. This O child could have a parent that's A and the other parent is B. That is totally possible because notice that A could be this IA little I. The B could be IB little I. And so notice that the genotype could be from these two, from A and B, and you might get blood type O. So even though neither parent is O, they could still have a child that is O. And so this shows multiple types of variation in Mendelian genetics, codominance, because A and B are codominant, and multiple alleles. There are more than 2 alleles. There are 3 alleles, the A allele, the B allele, and the O allele. So question, in the ABO blood system in human beings, alleles coding for A and B proteins are codominant while the O allele is recessive. In a paternity dispute, a type O woman has a type A child. What possible blood groups could the father of the child have on the basis of this evidence? So what are his possible blood types? So pause your video, and when you're ready, push play. So if you worked through this problem, the answer is blue, type AB, or type A. So if the child is type A and the mom is O, mom can only give a little I, which means that the child had to get the A from dad. So dad has to have an A allele. Dad could be AB or dad could be A. Either one is a possibility because both of those could give the child an A allele. Dad can't be O because if dad is O, that would be a cross of little I, little I, little I, little I. All the children would be O. There's no possibility of getting A. Dad can't be B because the child got the A allele from dad. So the answer is blue, type AB, or type A. So the next variation in Mendelian genetics is what's called pleiotropy. And that is where one gene affects more than one phenotypic characteristic. So there's not just one phenotype that comes out as a result of this one gene. So pleiotropy basically means one gene, many phenotypes. So an example of this would be sickle cell anemia. Sickle cell anemia is due to a mutation in a single gene. It's a single mutation in the beta globin gene. Yet, the sickling of the red blood cells creates a whole cascade of symptoms. Pain, stroke, vascular systems, kidney problems, brain damage, rheumatoid, pneumonia, paralysis, a whole host of different types of phenotypes that happen from this one gene being mutated. And so that's pleiotropy. One gene, multiple phenotypes. And so that is, you're going to see in contrast to when we talk about the next one, polygenic inheritance, where polygenic inheritance is multiple genes coding for one phenotype. Pleiotropy is the opposite. One gene, many phenotypes. So polygenic inheritance, multiple genes control a single phenotype. So examples of this would be things like height, right? If you think about height, are there only 2 phenotypes in the population? No, there's a whole host of heights, right? Height varies quite a lot, and it just depends on which combinations of genes you might get. So if you cross somebody, let's say that is big A, big B, little b, big C, little c, that would be kind of an intermediate height. So all of the capital letters would give height, would give tallness. So the A, the B, and the C, the capitals, give the height. The little a, little b, little c gives not a lot of height. So if you have half, so 3 out of 6 are tall, that person would be intermediate height. Now, if you cross 2 intermediate heights, intermediate height is the most likely. You're most likely to get around that intermediate, but very tall and very short individuals are possible as well, because it simply comes down to, by chance, which genes are inherited. So my family has a good example of this. Both of my grandparents on my dad's side of the family, neither one was especially tall. My dad is 6'2". His sister is 4'11". So by chance, my dad happened to get all of the tall alleles. His sister got all of the short alleles. And he had 3 siblings in between that were in between in terms of the height spectrum. But you can see that you could have 2 people with intermediate height, and you could have children that are either very tall or very short simply by chance. And so the range of phenotypes forms what we call a normal distribution. You would expect that the most likely would be to be intermediate height, but you could have very tall or you could have very short just by chance. Skin color is also this way. Skin color is also polygenic, as well as influenced by environmental factors, which we'll talk about in a minute. But skin color is also polygenic, right? If you think of skin colors, there's a whole host of different skin colors. It ranges quite a bit because there are multiple genes controlling skin production, skin color, pigment production. And so it's dependent on which alleles you have to get. So if you think back to the beginning of this lecture when you saw the twins and they had parents that were intermediate, they had one child who had darker skin and one child who had very fair skin. It just so happened the one with fair skin happened to inherit more of the fair skin alleles, simply by chance. You don't expect that to happen very often, but it can happen. It is a possibility. And so that is polygenic inheritance. And so next we come to environmental impact. And so if you think about, there's a debate, right? Nature and nurture. Nature means that your genetics plays a role. Nurture means that environment plays a role. Some traits are not influenced by nurture, meaning there's no environmental component. So for example, blood type, right? That is not influenced by environmental factors. That is simply a genetic thing. But certain traits can be influenced by environment. Skin color would be an example. So notice that these are identical twins. One twin has darker skin than the other, simply due to going out in the sun more, because sunlight is going to increase the production of melanin. Susceptibility to diseases such as cancer has both hereditary and environmental components, right? UV exposure can increase your risk, as well as there being genetic component to it. The color of hydrangea flowers. So these are hydrangea flowers, and depending on the pH of the soil, even though genetically these can be the same, if you plant them in different pH soil, the color of the flowers will be different. And so the color of the flower is dependent on the environment, meaning the pH in the soil. So here is a review for you to practice your genetics. You don't need to post this on Canvas. However, I'm going to wait and I'll give you the answers to this when I give you the answers to the other genetics problems. But I would like you to practice this on your own. See if you can review what terms you would put in this chart. So like A, for example, genes. Alternative versions are called blank. What do we call the alternate forms of a gene? Put that there, etc. So that's what you're going to do for this part. So now we're going to look at sex-linked genes, genes that are located on the X chromosome. Humans perceive light through the activity of specialized cells, rods, and cones in the retina. Different sets of cone cells are sensitive to different light wavelengths, so different colors. So rods perceive light intensity, colors are for cones colors. Now we can have mutations that affect the ability of subsets of the cone cells to register light, and that will affect the ability of an infected individual to perceive color, meaning that if you have certain genes that are mutated, it doesn't allow you to perceive color correctly. 5 to 8% of Caucasian men are colorblind, but only 0.5% of women are born colorblind. So notice that men are 10 times more likely than women to be colorblind. And so why is there such a difference in the incidence of colorblindness between males and females? And so here is an example of a type of colorblind test. Now don't panic if you can't see these correctly. Sometimes the screens adjust the color and it might make them harder to perceive. But if you look, the one on the left, there is an 8. So here is your 8. Now, if you have a slight form of color blindness, you might have perceived this as being a 3. Like you could see the really red ones, but the ones that were a little more orange might have been more difficult for you to see. And you might have thought that that was a 3. This is another example. So here we have a square, and within that square, we have a star. And so if you couldn't see the distinction between those colors, then you would have color blindness. So why do we have this incidence of males having this trait more likely? Well, we need to go back and talk about our sex chromosomes. So sex chromosomes determine sex, right? Determine male or female. X and Y are not homologous chromosomes because remember that homologous chromosomes are matched in size, in banding patterns, and the genes that they contain. Here is an electron micrograph showing those. And so you can see that the X, that's on the left, the X is much larger than the little tiny Y. So X is much larger than Y. Now, although these are not homologous chromosomes, during meiosis, when homologous pairs line up at the metaphase plate in metaphase 1, the X and the Y will behave as though they are homologous pairs. So they will line up side by side, and then they will assort independently. Gametes will only carry one of the two. So the gametes are only going to contain either the X or the Y. Males are XY. Females are XX. Now, if we look at the types of genes that are on those chromosomes, so the X chromosome contains between 8 to 900 genes, and the Y has about 50 to 60 genes, according to the National Institute of Health. So notice that the X has a lot more genes than the Y. What determines sex, however, is that there is a particular gene on the Y chromosome, and this gene is called the SRY gene, and that's for sex determining region. So it's this specific part of the Y chromosome specifically that dictates maleness. Now, interestingly, female is pretty much like default. By default, babies would be female. It's the presence of that SRY, that gene on the Y, that has them produce more testosterone and makes them male. So there have been instances of a child being XY, so they should be genetically male, but phenotypically they're female. And that's because that Y chromosome has a mutation in the SRY gene. It did not turn on production of testosterone, and by default appears more female. So that's just something that's kind of interesting about X and Y. Now, most sex-linked genes are X-linked, simply because if you look at the X chromosome, there are a lot more genes on that chromosome than there are on the Y chromosome. So it's much more common for sex-linked genes to be on the X chromosome. Now, if we think about who determines the gender of the offspring, who determines the gender of the offspring? Well, think about it. A female is XX. What that means is she is always going to give her offspring an X chromosome. That is all she has to give. Now, males are XY. So when sperm goes through meiosis, right? So if we look at the adults, so here's a male, 44 autosomes plus an X and a Y. So when that goes through meiosis, 22 autosomes and an X, or 22 autosomes and a Y. Depending on which one fertilizes the egg, that's going to determine the gender or the sex of the offspring. So if we have our egg, right, and our egg has X, and your textbook had a little problem with its image. Notice that they had the female being XY. Females are not XY. Females are XX. So the female should be XX and 44 autosomes. And what that means is that she only has one type of gamete, 22 autosomes, and an X, not a Y. That should not be a Y. So if the egg has the X and the sperm has the X, the offspring are going to be female because XX is going to give females. If the egg has the X and the sperm has the Y, the offspring are going to be male. And so it is not the female that determines gender. It is, in fact, the male that determines the gender, because it depends if the male provides sperm that are X or Y. And so in England in early days, kings would get upset when their wives wouldn't give them sons, not realizing that that was on them. That was their fault, right? Their wife had no bearing on whether she gave him a son or a daughter. That was all the male's choice by the X or the Y in the sperm. Now, for X-linked genes, the reason that it's more common in males than it is in females has to do with the fact that males only have one X chromosome. They don't have a homologous pair, right? Because males are XY. So in order for a male to have the trait, he only has to inherit one bad copy. Females have to inherit two bad copies. So let me explain this. So let's say we're looking at hemophilia. Hemophilia is a disease where people who have the disease have problems with blood clotting. Their blood fails to clot properly. So hemophilia is sex-linked recessive. It's carried on the X chromosome. So for males could be X little h, meaning they have the hemophilia allele, and Y. That would lead to an affected male because it's homozygous recessive, right? Or in this case, what we call hemizygous. They have half of it. The hemizygous recessive means that they're going to have hemophilia. They're not going to make any of that normal clotting factor. If the male is X big H Y, that is an unaffected male. He's going to blood clot fine. Now let's look at the possibilities for females. X big H, unaffected female. Because she has a big H, she doesn't have hemophilia. X big H, X little h is an unaffected female, but she's a carrier. So she doesn't have hemophilia, but she does carry it because she still has a good copy. X little h is an affected female. She would have hemophilia. So notice that the only way you get a female that has this sex-linked disease is if she inherits 2 bad copies. Males only have to inherit 1 bad copy. 1 bad copy is enough for them to have that trait. This is why it's much more prevalent in males than it is in females. So now let's think about where those alleles come from. So if you have a male and the male has hemophilia, who did he get that trait from? Was that trait inherited from mom or dad? So think about that, pause if you need to, and then push play when you're ready. The answer is if a male has the disease, he got it from mom. Because notice if a male is XY, the dad gave the son the Y. The X had to come from the mom. So if a son has hemophilia, he inherited it from his mom. His mom could either be a carrier, right? Meaning she doesn't have hemophilia, but she carries it, or his mom has the disease. So that trait comes from mom. Now let's think about daughters. Let's say we have a female who has hemophilia. If a daughter has hemophilia, who else in her family definitely has hemophilia as well? Answer, her dad. Because the only way she gets to be X little h is if she got a little h from both parents. That means that dad was X little hy. Mom could have hemophilia. She could also be X little h, or mom could be heterozygous and be X big H, X little h. But if we have an affected female, we know that dad is also infected, also has the disease. So this is just showing some different examples of how this can be inherited. So in this case, let's talk in terms of color blindness, which is also X-linked recessive. So we could have a female, and the female is X big N, X big N. So X big N, X big N. That female is not colorblind. She has normal vision, and she's not even a carrier. And that woman has a child with a male who is colorblind. So a male who's colorblind and a female who is normal. This male has 2 choices for gametes, X little n or Y. This one, the only possibility is X big N. So notice when we fill in our Punnett Square, there's a 1/2 chance that they could have a daughter that is a carrier, or a 1/2 chance that there's a normal male. Notice at this point, there's no possibility of having a child that is colorblind. Now, let's say we have a female who is a carrier. She is X big N, X little n, and a normal vision male, X big N, Y. So if we fill in our Punnett Square, X big N, Y, X big N, X little n, this is the mom that's a carrier. The probability of having a colorblind son, 1 out of 4, right? Females, there's no possibility because dad has normal color vision. Now, this would be my scenario. My dad is colorblind, so I know I have to carry it because I know my dad gave me the X little n allele. He was definitely colorblind. So I'm a carrier. My children's father is not colorblind, so he would be this. So again, when questions are asked about this, you have to pay attention to the wording. So if I gave that background information that I am not colorblind, but my dad is, and that my ex-husband was normal vision, and I said, what is the probability that they will have a colorblind son? That would be 1 out of 4. Will have indicates they don't have a child yet. What if I asked it in a different way? And I said that I have a son, and my son, what is the probability that he is colorblind? So my son is already born. We know he's not either of these possibilities. Those are now gone. He's already born. He can't be those possibilities. So the probability that my son is colorblind is 50-50. He had a 50% chance of being colorblind. Lucky for me, he's not. He has normal color vision. But there was a 50% probability once he was born that he was colorblind. Now, again, the only way that you're going to end up with a colorblind daughter is that mom is a carrier, dad is colorblind. Only when they both have an X(n) to give can you see a daughter who has the disease. So, question for you. Red-green colorblindness is caused by a sex-linked recessive allele. A normal vision man marries a woman with normal vision whose father was colorblind. What is the probability that they will have a colorblind daughter? And the next one is, what is the probability that they will have a colorblind son? So work through that, push play when you're ready to go. So let's work through this. So we have a normal vision male, that's this square that's not filled in, marries a woman with normal vision whose father was colorblind. So now we need to go in and fill out that the males are all XY to start, and the females are XX. All right, so let's use the letter N for normal vision. So if the male is normal vision, he is XY. What allele is he going to have on his X chromosome if he has normal vision? Answer is he has to have a big N. Now, the female, she has normal vision, so we know she has a big N, and then something on her second allele. Notice her dad is colorblind, which means that her dad has the recessive allele, so her second allele has to be the recessive. So she is X big N, X little n, because again, dad was X little n, Y. So now we just fill in our Punnett Square using those genotypes. So here is our Punnett Square. So notice this man is normal vision, so X big N, Y. Female is a carrier. She is X big N, X little n. So we fill in our Punnett Square. X big N, X big N, X big N, Y. X little n, X little n, X little n, Y. So what is the probability that they will have a colorblind daughter? That should be a big N. So they have a 0% or no possibility of having a colorblind daughter, right? Because here are the girls, none of them are colorblind. Now, the next question says, what is the probability that they will have a colorblind son? So what is the probability that they will have a colorblind son? So there is the son that is colorblind. So the probability that they will have a colorblind son is 1 out of 4. So red for the daughter, 0, and 1/4 for the boy, the colorblind son. Now, there's a biological problem. Females have 2 X chromosomes, and each gene on the X chromosome should be expressed at twice the level as it is in males. Because if you think about it, if she has 2 X's, each of those genes is expressed twice as much as it is in a male. And so how could one avoid this problem so that genes on the X are expressed at equal levels in males and females? And the answer is that there is this phenomenon called X chromosome inactivation. In female mammals, one of the 2 X chromosomes is highly compacted and transcriptionally inactivated. So what that means is it packs in really, really tight, and it forms a structure called a bar body. And when the bar body is packed in tight and those genes are packed in real tight, we can't turn on gene expression because RNA polymerase can't get access to the promoters to initiate transcription. Now, this process is random. There is random inactivation of either the maternal or the paternal, and it happens early on in development. Some cells inactivate the one from mom. Some cells inactivate the one from dad. Now, of course, this still only happens in females. This phenomenon doesn't happen in males because males are XY. They don't have two X's. This only happens when we have two X's because that would allow for only half the amount of genes to be expressed. We only want one of the two X to be expressed. So an example of this would be tortoiseshell fur coloration in cats. So in female cats that have a black allele and an orange allele, they get this tortoiseshell pattern, this black and orange patches pattern, because of X inactivation. And so let's take a look. So if we look at the female tortoiseshell cat, so on her X chromosome is the allele for fur color. If she is heterozygous, she has an orange allele and a black allele. Now, early in development, each cell will inactivate randomly. So they will inactivate randomly. Some cells, the black will become inactivated. That one's going to pack in and that's going to form the bar body. And the orange allele is going to be expressed. And so you get these patches of orange fur. In other cells, the X chromosome with the orange fur is inactive and the black fur is the one that's being expressed. And so this genetic modification, this changes to the DNA structure, not the sequences, but actually the structure of the DNA is what is going to have this effect. And so you get these cats that have these patches of orange and black fur. And even if you have identical twins, even if you have clones for a cat, even though they're genetically identical, physically they might look different because of their X inactivation, because of the random nature of inactivating the X chromosome. Genetically, they could be totally the same, except which X is activated could be different, which leads to different phenotypes. And so that is something that we will talk about later about epigenetic changes. So changes to DNA structure, which affect gene expression. And that's how you can have twins that while genetically identical, they might have phenotypical differences because of their chromosome structure. This is seen in the case of astronauts. There's a pair of twins that are astronauts, and when one went and lived in space for a period of time and then came back, it was definitely noticed that when they checked the pattern of the DNA methylation, which controls gene expression, was now very different between the twins. So that environmental influence affected that and led to more differences between the twins. And so there's a video that you're going to watch about epigenetics and about how DNA structure affects gene expression. But this is X inactivation. This is talking about the X chromosome specifically. So an example of X inactivation in humans is something that's referred to as ectodermal anhydrotic dysplasia, or EDA for short. And in this condition, there's abnormal development of skin and associated structures such as hair, nails, teeth, and sweat glands. And so in females that have this condition, she will have a mosaic appearance. And what that means is that this gene is on the X chromosome, and randomly, early in development, we get random inactivation of the X chromosome. And so all the cells that derive from that cell will have the same X inactivated. And so she's going to have regions where she is affected. So that's this kind of yellow color here. Those are the cells that are affected. And in the cells that are affected, they lack sweat glands. The anhydrotic is referring to without sweat glands. So they lack sweat glands in those regions. They have skin abnormalities that have areas that are dry, wrinkled, or darker in color than the surrounding skin. Individuals tend to have sparse scalp and body hair. They might have missing teeth or teeth that are small and pointed. The reduced ability to sweat because they have fewer sweat glands. And so a problem with having the inability to sweat is that that can lead to dangerously high body temperatures, particularly in hot weather. And so they lack the ability to cool down properly because of these missing sweat glands. And the severity of this is just depending on random which X chromosome is inactivated in which cells. So another example in terms of X inactivation. So remember that I said for color blindness for females, if a female is going to be colorblind, she has to be X(n), X(n). However, there is an exception to this. And that is that remember that the X chromosome is going to undergo random X inactivation. So if you have a female who's a carrier, she carries the colorblind allele. In some of her cells that lead to her cones, she might have her good X chromosome being inactivated and her colorblind allele being expressed. And so in that case, she might have a milder form of colorblindness. She might not be fully colorblind, but she might have a more difficult time distinguishing between closely related colors simply due to random X inactivation. It might be that her good X is the one that inactivated. And so this is looking at some X inactivation in humans.